All right, hello everyone. Welcome to tonight's event. All right, we're gonna get started. So I wanna welcome all of you to tonight's lecture, and I'll be introducing all of our amazing lineup. So let's get started. I'm Dr. Diana Lee. I am the Associate Director of Education and Training Initiatives at Columbia University's Zuckerman Institute, where researchers from departments across the university all come together to transform our understanding of the mind and the brain. And we're extremely pleased to continue sharing this incredible research through our public engagement programs focused on science and the brain, including tonight's event, which is part of the Stavros Niarchos Foundation Brain Insight Lecture Series. So with the Foundation's generous support, we continue our commitment to exceptional science and outstanding programming that highlights the ways that neuroscience intersects with our lives every day. So each lecture in this series provides us with a really special opportunity to spotlight world-class researchers at the forefront of their fields addressing important scientific issues to inform and engage our community. And we hope tonight's event will give you a unique view into that and leave you inspired by the research taking place at Columbia University at our Zuckerman Institute. So closely paired with this lecture series is the Stavros Niarchos Foundation Teacher Scholar Program, which engages a really amazing cohort of 24 science teachers who bring this content from these lectures into their classrooms across New York City. And it's a joy to continue welcoming our teacher scholars locally in person to this lecture series. And we're also really grateful to see so many of you tuning in online right now from across the world. And in particular, I would love to thank the Stavros Niarchos Foundation and the foundation members who join us here tonight for their continued partnership in our programming and their resounding commitment in helping us make brain science accessible for all. So tonight's lecture is all about how the cells in the brain choreograph movements we make, from maybe the simplest tapping of a finger all the way to the most sophisticated learned behaviors that we usually delight in seeing on concert stages and at the Olympics and all of those amazing televised events. And personally, I'm pretty thrilled about today's event, and maybe I'm biased because my own PhD research was on how the neurons in squid give rise to their high-speed swimming and jet propulsion, but maybe we all agree that this is a really fascinating topic, and I'm especially excited to hear from today's neuroscientists who study movement in different types of animals and learn what this work can teach us about the brain behind the behavior. So, our experts in today's event will treat us to presentations from two incredible speakers, and that will be followed by a conversation between them led by the Zuckerman Institute's own Dr. Sumaira Zamarad. So Dr. Zamarad is a neuroscientist and associate research scientist in the lab of Dr. Richard S. Mann at our Zuckerman Institute. And she was born and raised in Pakistan and landed in the US to earn her bachelor's in biochemistry at Stony Brook University, followed by her PhD at the Albert Einstein College of Medicine in the lab of Julie Seacombe. There, she studied intellectual disability mutations in Drosophila, the fruit fly model organism. Currently, Dr. Zamarad is investigating the intricate neural circuitry that is responsible for eliciting specific walking behaviors, once again in her favorite model organism, the Drosophila fruit fly. She is the recipient of the Brain Initiative Ruth L. Kirschstein F32 Postdoctoral Research Fellowship. So, before I welcome her to the stage, if you folks are watching online, I encourage you as you listen to our event to use the Q&A function at the bottom of your screen to submit questions. And for our in-person folks, please raise your hand during the Q&A portion of our event, and Dr. Zamarad will invite you to ask your question by speaking into a microphone so we can all hear you. So please join me in welcoming our host and moderate, Dr. Sumaira Zamarad.
Thank you so much, Diana, for your very kind introduction and inviting me to moderate tonight's event. Thank you to SNF for having these very engaging events. In today's event, we will be hearing from Dr. Gwyneth Card and Dr. Mark Churchland, two experts who study the neuroscience behind movement. Our speakers will discuss how the brain's complex network of cells work together to orchestrate the right action at the right time. This question is also very intriguing to me myself throughout my postdoc research here at Dr. Richard Mann's lab, where I too have been studying the neural circuitry that is important to regulate various very precise walking behaviors in Drosophila, fruit fly. So it's very amazing to see how there's so many multiple interconnected neural pathways, so lots of different neurons connected in what we call circuits, that are involved in successfully carrying out these motor movements in limbed organisms. And all of this is very essential for navigating our complex, ever-changing environments in our lives and also for our survival. And you can just imagine that if any of these circuits or even subsets of neurons are dysregulated, they can have varying degrees of devastating effects in our ability to function fully. And our work and research here at the Institute, and of the great neuroscientists who are going to join us today and tell us about their work, is basically going to circle around our basic and fundamental understanding of how these neurons can work in these highly orchestrated and well-synchronized circuits, and how that's crucial for understanding eventually how they may have an effect in regulating health and disease. So I'm very excited to have this opportunity for all of us today to learn about how our nervous system accomplishes these important tasks from the experts in the field right here at our institute. In today's event, you're going to hear two 15-minute talks, one from each speaker, and then I will moderate a discussion in which we will include questions from you, our audience. If you have already submitted a question, thank you very much. If you wish to submit a question while the talks are in progress, please look for the Q&A button to submit your questions to the panelists. And please do let us know if you are a teacher or a student. If you are a student, tell us what grade you're in. We mentor students all the time, so we love questions from curious students. And now it is my great pleasure to introduce our first speaker, Dr. Gwyneth Card, who studies how neurons function as the building blocks of behavior. So Dr. Card is an associate professor here at Columbia University's Zuckerman Institute, and also an investigator at the Howard Hughes Medical Institute. Her research aims to understand the neural mechanisms and circuit architectures that underlie behavior choice for ecologically relevant, visually guided behaviors of the fruit fly Drosophila. Her recent work has focused on using automated, high-throughput behavior tracking together with genetic tools and electrophysiology in behaving flies to study the neural circuits that govern simple decision behavior during Drosophila escape responses. No easy task, I tell you. Previously, Dr. Card was a group leader at the Howard Hughes Medical Institute Genelia Research Campus. She received her PhD with Dr. Michael Dickinson at Caltech, an MPhil with Dr. Simon Laughlin at the University of Cambridge, and a bachelor's degree from Harvard University. And I'd like to add that Dr. Card was a part of some very foundational work at Genelia, where she was working to identify and generate tools for neurons called descending neurons. And she also worked on the Drosophila EM dataset, which we might hear about today. And I have heavily relied on these tools in my research as well, along with many other researchers. So please join me in giving Dr. Card a warm welcome to the stage.
Thank you so much for that introduction. It's a real pleasure to be here today with everyone. Thank you for coming out to hear a little bit about our science today. So our bodies are capable of innumerous different motions, right? From playing tennis to running down the street to picking up our kids or our pets. And what my lab has been interested in for the last decade or so is trying to understand how it is our brains, which orchestrate these motions, choose the right action to do at the right time. And we're really interested in trying to understand this at a mechanistic level, the level of individual brain cells. How is an individual brain cell, which is called a neuron, contributing to this kind of processing? And this involves taking in sensory information from our world around us. What are we seeing? What are we hearing? And processing that together with internal information. How are we feeling? Are we hungry? Are we sad? Are we tired? In order to decide what action to do next. And what I want to do today is I want to give you a little bit of a flavor of how we go about trying to go from these external observations of movements that we make and movement choices that we make to understanding these internal workings at the level of an individual neuron. And so I'm going to focus on a particular question that my lab has been looking at recently. That's a very important one for this kind of understanding how movements are generated. And it starts with this object that many of you are probably familiar with. You've all encountered this exact problem on a daily basis. You wake up in the morning, you're thirsty, you want your cup of coffee. It's sitting there on the table. Well, in order to get that cup of coffee, you first have to see where it is. Is it in front of you? Is it to your right, to your left? And then your brain has to direct your arm to actually reach towards the cup and pick it up. And even though you may not realize it, your brain is doing something fairly sophisticated in this case. It's actually transforming information that you see on your eyes about where the cup is in your visual space into commands that have to be generated in some kind of language of your body, some kind of motor space. Should I extend my arm? Should I contract it? Should I reach it this way relative to my body? And so this is called a sensory motor transformation, and how this transformation works underlies much of the movement that we do. And so we're really interested in trying to unpack what's going on in the middle there. Okay, so to do that, we don't study humans. We study this creature you heard a little bit about, Drosophila melanogaster, the fly. The first thing to know about the fly, if you are not so familiar with them other than trying to swat them, is that flies do in fact have brains. So this is a view of the back of a fly's head. The back of its head has been cut open by a scientist, and we use genetic tools to drive fluorescent proteins, in particular neurons we're interested in. This is why we like the flies as a model organism. And so you can actually see individual neurons glowing in this fly's head. Okay, so that's one reason to use the fly. The other reason is that in addition to being able to access these neurons individually, there aren't that many of them. We have some 86 billion neurons in our nervous system, in our brains. Flies, the whole central nervous system, is only about 300,000 neurons. If you can imagine, if what we want to do is interrogate each of these neurons one by one, that's the difference between having to, say, interview the number of people in 12 Earths compared to the population of Anchorage, Alaska. Right? So this is a much more tractable problem that we imagine might even be solved in the time scale of a single career. So it's very exciting for us. Now, there's one more important reason why flies can be a good model system, and that is that brains, whether it's a fly or a human or a mouse, are basically built from the same building blocks, and that is these brain cells, neurons. So this is what a human neuron looks like. It has a cell body. Around that, it has what we call dendrites, which are where inputs are received. And then the cell is going to integrate those inputs. It's going to send an electrical signal down to its axon to outputs, other neurons that it contacts. And that's how information is passed and transformed in general in the brain. Well, fly brains are also built of neurons. They're basically the same as ours. They have one key difference, which is that their cell bodies are not actually in the middle of their dendritic field. They're actually kind of float out like little balloons on the side. But other than that morphological difference, basically the same Lego pieces are composing our different brains. And so we hope that what we learn in the fly could in some ways translate to understanding our own brains. Now, not only are the pieces the same, but how they connect is the same. So neurons connect to other neurons by something we call synapses. And that is that after the electrical signal gets to the end of the axon, there's a little break to the dendrites of the next neuron, and we actually have to pass chemical information from one neuron to the next. And synapses are going to be an important theme tonight because how many synapses there are from one neuron onto the next tells us something about how much information is passed along the chain and how information may be transformed. Okay, so that sets some of the building blocks for why we're going to use the fly, but there's of course one problem. Flies don't actually drink cups of coffee. So how can we approach this type of problem in a fly? What kind of behaviors we can use? Well, here's where we turn to innate behaviors that are actually remarkably the same across different species. So for example, here's a person walking down the street. Now he's going to encounter an unusual situation, but you'll see he responds very impressively. And that is that an unexpected looming stimulus, so a looming stimulus is an object coming at you and expanding on your eye, comes at him in the form of a car up on the sidewalk. And as this replays, you can see that his escape reaction is basically to lean his body over away from that direction of the car and then push off. Well, we can recreate that in the lab with a fly. So in the lab, we make our own artificial looming stimuli. We basically take dark circles projected on a screen and expand them in front of the fly. And then we film fly reactions with high-speed videography. So this is a video that was filmed at 6,000 frames per second, because as you know from trying to swat a fly, they're very fast. Well, let's see what the fly does. Basically does exactly the same thing. It leans over away from the direction of the looming stimulus, it pushes off with its legs, and it takes off. In both these cases, exactly the same sensory motor transformation is having to occur. The visual system of the animal is having to detect where that looming is coming from, and it's having to translate that into some motor language to get the animal to go in the correct direction to safety. And what we've discovered by taking hundreds and thousands of these kinds of videos of the fly is that actually flies have really exquisite control over their escape direction. So here you're seeing 3 different experiments we've done. In black is the direction relative to the fly that we had our looming stimulus come from. And each green arrow represents one fly that we observed taking off and the direction that that fly took off. And then here, these heat maps are basically the averages of these arrows, and where it's pink, it's showing you kind of the average direction that the fly went. So you can see when the stimulus comes from over here in front of the fly generally goes backwards, vice versa. If the stimulus is from the back, it goes forwards. And if the stimulus is from the side, it sort of goes towards the other side with a little bit of a bias to go forward. Now, one of the secrets we discovered from looking in detail at these high-speed videos is that the way that the fly does this is actually exactly through that kind of postural shift you saw in the video. So when the fly sees a looming stimulus, it might initially have its center of mass, which is sort of the middle of its body, relative to its 2 jumping legs, somewhat randomly distributed. However, after those leans that the fly does, what we find is that now its center of mass is put in a very particular target location, such that here, as you can see in the color code, it's representing the direction the fly is going to take off. So if it sees a looming stimulus coming from its left side, it moves its center of mass over to its right side, and then that's going to indicate that it's going to take off to the right. And so these postural adjustments that the fly does that you saw in the video before its actual escape are key to setting this direction. Okay, that's the fly's behavior, but now how do we go into the fly's brain? Well, another reason that the fly is a great model organism at the moment is because we have a very special tool, and that is what we call a connectome. So what you're seeing here is the brain of the fly, and this is actually a volume of images collected from an electron microscope. And what that lets us do is get very, very high resolution images. So we've now zoomed far into the brain, and each of these kind of oblong, circular objects that you can see is a part of a neuron. And by going through each stack of this volume, little bit by little bit, we can trace those and connect them such that we can reconstruct the neurons in the fly's brain. And actually, we're now able to reconstruct all the neurons in the fly's brain. And Janelia has led a project to do this densely for about half the brain. And what you get as a result are you get these beautiful individual neuron morphologies, as well as we can see the synaptic connections between them and we can count them. And so we know where and how many connections there are between each of these neurons. And that lets us draw an entire graph. That graph is what we call the connectome of this connectivity. And so I'm highlighting these particular neurons because these are exactly at a location in the brain that's difficult to get to in a really, really large brain like ours, but that now with a connectome we can access, which is right in the middle between where sensory information turns into motor information. Okay, well, what do I mean by that? Well, this part of the brain over here is what we call the fly's optic lobe. And that's purely processing visual information. So it has photoreceptors in it that transduces light. It gets processed over a few synapses here. But then these sets of neurons in blue and cyan and in yellow are projecting out of the optic lobe, and they're now going into the center of the fly's brain. And what we discovered by looking at the connectome is that actually some of these neurons, which are carrying visual information because they're coming out of the optic lobe, connect directly to this type of neuron shown in pink here. And this type of neuron you can see has dendrites up here in the central brain, but then it has this very long axon that actually leaves the brain, goes through the fly's neck and down into the body. And we call those descending neurons. So in the nerve cord of the animal, which is where this neuron goes, are motor neurons that move the body. So this neuron is already talking to the motor system of the fly, and these neurons are coming from the visual system of the fly. And across this set of connections is exactly where they meet. So what can we learn about that? Well, first we wanted to know what these types of neurons that project out of the optic lobe, what are they actually responding to? And so to do that, we did a set of experiments using an indicator, something we can put into the fly's brain, into individual neurons, that lights up when the neurons are active. And so there's about 10 of these different kinds of visual neurons that we looked at. And here's the whole population of a particular type. We call these LC neurons. This is LC18. But we wanted to look at individual LC18 neurons. So in our microscope, you can see we can identify individual axons from them. And then we show a whole bunch of different visual stimuli to the fly while we're looking at our calcium indicator under the microscope. And these are average responses that we get from these different cell types. And so you can see one take-home message is that different types of visual neuron respond to different types of visual stimulation. Some of them respond to bars, some to gratings, et cetera. But there's a whole set here that respond to exactly the stimulus we're interested in, looming. And in particular, there's one called LC4, one of these LC neurons that responds to looming, that we found from the connectome connects to a whole range of descending neurons. Okay, well that made that particular set of descending neurons of interest to us, because remember, these descending neurons now connect to the motor system. So we wanted to survey the descending neurons. We can use another type of experiment called optogenetic activation, where we use gene manipulation in the fly to express a channel in these descending neurons that when we shine light on the fly, it opens and it activates the neuron. So we can turn on these neurons with light. And when we do that in 2 of these particular neurons, here's what we see. So here's a whole range of different flies. When we activate DNP11, what you can see is that these flies all lean forward. You can see now that the green fly that was the first position of the video, you can see the ends of the flies, meaning it's moved forward. We found a second descending neuron, which when we activated it, now the flies all lean back. So what you're going to see is you're going to start to see heads appear as these flies lean back. So now we've found looming neurons that connect directly to these posture-changing neurons, one that leans the fly forward and one that leans the fly back. And I'm showing you example videos here, but of course, it's good science to quantify what we see, and so we did that across many, many flies. And so here's the demonstration of the leaning forward. Here's DNP11 when it's activated. If we measure the angle between the fly's jumping legs and its center of mass to see how that changes with the lean forward or the lean back, DNP11 activation makes the fly lean forward. DNP02 activation makes the fly lean back. And in fact, if we let the videos play, those postural adjustments lead to actual takeoffs. So here, DNP11 activation makes the fly take off forward, and DNP02 activation makes the fly take off backwards. Okay, so we have some sense of how these are hooked up, but that's not quite enough to really understand how the system works. And to understand those details, we now had to turn to the specific connections between those visual looming detecting neurons and these descending neurons that drive the animal in a particular direction. How are they actually hooked up? And the first secret that we discovered, again, turning back to the connectome, was that in fact, these LC4 neurons, the looming detectors, of which there are about 50, they look in all different directions. So there's some LC4 neurons that detect looming over here, some over here, different parts of the fly's visual field. Individual LC4 looming detectors don't actually have the same number of connections onto a given one of these descending neurons. If we look at each individual LC4 neuron and how many synapses or connections it makes with DNP02 that leans the fly back, you can see sometimes it's almost none and sometimes it's up to 50. And then when we look at exactly these same looming detector neurons and how they're connecting onto our forward-leaning descending neuron, we found that in fact it was just about the opposite. Individual looming detectors looking at a particular point in space that make a lot of connections with the lean back neuron make very few connections with the lean forward neuron, and vice versa. And so what I'm going to show you now is a visualization of that. If you imagine that we're now going to color code this number of synapses from blue to red, with red being a lot and blue being not very much, and we're going to look at how that's distributed across LC4 dendrites looking at different points in space, this is what we see. We see a gradient. And how is that gradient oriented? Well, DNPO2, remember, is the descending neuron that makes the fly lean backwards. And this hot spot here in red with a lot of connections to DNPO2 happens to come from the looming detectors located in the front of the animal's eye. And we see the converse in terms of the spatial pattern for the DNP11 that makes the fly go forward. So it makes sense, you could imagine, that if the fly sees looming from the front, the brain is actually hooked up to then drive the backward jumping neuron. And we can actually understand that very precisely mathematically. I'm just going to show you a little bit of a cartoon of that now, and Mark will get into it a little bit more. But you can imagine that we can actually take these vectors, which are the synaptic weights between LC4 and these two descending neurons, and this synaptic matrix, weight matrix, is actually representing how we're going to transform information as we go from activity in these neurons and try to predict the activity in the downstream neurons. And again, in cartoon form, what that would look like, so here's our gradients of connectivity. If we see a looming stimulus coming from the front of the fly, right, that's going to particularly activate the DNPO2, the one that makes it go backwards, because you have this hot spot here. Whereas if you see looming stimulus coming from the back of the fly, then it's going to activate this other gradient, which particularly stimulates DNP11, which is the one that makes the fly go forward. And so this kind of synaptic single cell resolution transformation is what's going on at various different scales of complexity in your brain to convert information from the world into actual actions. Okay, and with that, I just want to wrap up by showing you pictures of some of the folks who actually did the work. Science is a team effort, and we're always grateful to all the people that we get to work with. We get to work with some really outstanding scientists. In particular, I'll just point out to you Nathan Klopotki, who did a lot of the calcium imaging, Hiro Namiki, who's the hero that made a lot of these genetic driver lines for the descending neurons, and Katie and Jan, who did a lot of the electrophysiology that was in the background of this work that I didn't talk about today. All right, thank you very much.
Thank you so much, Dr. Card, for that fascinating talk. I know we learned a lot. Remember, if you have a question, please use the Q&A button, and we will try and answer them in the Q&A after the second talk. It now gives me great pleasure to introduce our second speaker, Dr. Mark Churchland, who is a neuroscientist specializing in how the brain prepares to act. Dr. Churchland is an associate professor in the Department of Neuroscience here at Columbia University, and he is the co-director of the Grossman Center for the Statistics of Mind. He received his BA in mathematics and psychology from Reed College in Portland, Oregon. He received his PhD in neuroscience from the University of California, San Francisco. His postdoctoral work was in the Neural Prosthetic Systems Lab of Professor Krishna Shenoy at Stanford University. Dr. Churchland's lab focuses on how the brain controls voluntary movement. Dr. Churchland is a recipient of the 2012 NIH Director's New Innovator Award. He received a 2015 Klingstein-Simons Fellowship Award, a 2013 McKnight Scholar Award, a 2013 Sloan Research Fellowship Award, and a 2012 Searle Scholars Award. I have more.
Skip it.
He was a 2006 recipient of the Burroughs Wellcome Fund Career Award and a 2003 recipient of the Helen Hay Whitney Research Fellowship. So lots of awards, all of them. But please join me in welcoming Dr. Churchland to the stage.
Thank you. All right. I'm pretty excited to go after Gwyneth. We had a good time putting this together. We got a little excited about matrices, so you're going to see a lot of matrices. We're kind of excited too about there being teachers and students in the room, and well, we're kind of overly didactic people ourselves. I didn't mean to be saying any of this, but I'm up here, so it's just it's just flowing. But I think also, like, despite having a degree in math and working with a lot of people who had a degree in math, we all felt like kind of confused by matrices and linear algebra. I don't know if you remember being taught this stuff in math class, like probably senior year. But you're like, what the? And the determinant? Like, what's the point of that? And I'm only really going to be able to give you a flavor here, building off of what Gwyneth did. But a lot of that stuff that seems weird when you first learn it, and honestly, it did for me even in college, a lot of that math makes a lot more sense in the context of neuroscience, and maybe 20 years from now, that's actually how they'll teach it in high school. It would probably, in all honesty, make it a lot easier to understand. Oh, sorry, I need to go back. And I'm already getting ahead of myself here. So I'm at sort of the opposite end, or nearly at the opposite end of the small and not human-like to large and human-like spectrum. I study principally rhesus monkeys, and I'm particularly interested in a brain area that they share with us, which is called the motor cortex, which is the part of the brain that controls essentially all of your voluntary movements. And a relevant point that will come up later, because some of what we're trying to do is to treat people to whom this might happen, is if there's damage to your spinal cord and your motor cortex can no longer talk to your muscles, you are paralyzed, right? And that's true even though your motor cortex is still working perfectly normally, it's still sending out perfectly normal signals. They're just falling on deaf ears because of the damage in the spinal cord. And that's not an uncommon thing to happen to people. We train monkeys to perform a sort of a large variety of video game-like tasks. I'm just going to show you one of them. Concentrate first on the left. So what the monkey is doing is he's pedaling with his arm in order to move forward through a virtual environment. There's a rule too, when it's green, you have to pedal forwards to move forwards. And when it's a desert landscape, you have to move backwards to pedal forwards. And he knows that every time he stops on one of those glowing squares, juice will flow. And he likes juice, so he stops on the glowing squares. And then when the juice stops, he knows it's time to move on to the next square. And as you can see, he's very good, he's very accurate. And then the kind of fun thing that's going on here on the right is that the pedal actually no longer does anything. It's been completely disconnected from the video game. He doesn't know that. And in fact, all the motion through the virtual environment is now created by our best guess as to what he's trying to do, recording neural activity from motor cortex. So you could imagine, and in fact this is done in a very small number of human patients, that you could use this to treat somebody who for example, had a spinal cord injury. So what does neural activity look like? Well, it looks like this. So every one of these lines, every one of these, this is about maybe 10, 20 milliseconds worth of data, so not much. Every one of these lines is the voltage coming off of a single electrode that's in the primary motor cortex while the animal's actually doing something. It would look much the same if it were in a person. And these events that you see, sorry, I don't really have a pointer that works. There we go, that works. These events that you see here and here, those are those spikes that Gwyneth was talking about. That's when the neuron becomes active and sends a message to all the other neurons that it connects with. And we can record at this point, and we're getting better, we can record a lot of electrodes simultaneously. We can record a lot of neurons simultaneously. This is actually slightly underselling the state of the art at the moment. And you can see here another example of what I was talking about earlier, right? There's this little event here that occurs the same way every time. And if we zoomed in, you'd see it really was the same way every time. And that's the repeated spikes of one neuron that in this roughly 100 milliseconds, so that's 1/10 of a second, fires, I don't know, what is that, 6, 7 spikes? You can count them as well as me. So we can turn all of this very complicated data into something just as simple as for every neuron, when did it emit a spike, right? And we end up with data that looks like this. So this is a very common way of presenting data. Every little tick mark is when a spike happened, and every row is one neuron. So this is about 200 neurons. This data was a few years old. We can do a little bit better now. And what you see up at the top there in the green is our best guess as to what the monkey is trying to do at each moment, right? And we're being honest here too. We're predicting the present. If you look at the far right, that's like now. You can think of that as the present moment. We have to predict that based only on data now and data in the past, right? We're not allowed to look into the future because if you're trying to help a human, you can't look into the future. But we can do pretty well. And so when he's cycling forwards, that's when you see those vertical deflections. That's positive rotational speed, and you'll see it go negative in a little while as well, and we're going to track those as well. And very importantly, also, we can tell when he's trying not to move. You can imagine if you were a patient and this was supposed to be, you're supposed to be controlling a wheelchair, the last thing you want is for it to move when you don't want it to. So normally this is in here because you're like legally required to tell people. This is more at this point just kind of a fun way of noting that we really are hoping that this will help humans at some point. And the amount of money we plan on making off of this is like literally hundreds of dollars. So it's like kind of an embarrassing financial disclosure in all honesty. The real goal is to help people. So there's a cartoon on the left. I don't want you to try and understand it at all. We're going to understand it a little better at the end. For right now, kind of treat it like art. That little ball there, I'm going to pop back once, I'm going to pop forward again so you can watch this. That little ball is our best guess in some very abstract way of what the brain is doing at each moment. And when it's going around the purple track is when the monkey's cycling forward, or at least when we think he's cycling forward, and when it's going around the orange track, that's when he's cycling backwards. So if we know what neural activity is doing, we can make our best guess as to what it is he's trying to do, which is sort of the whole game here. So, and we'll understand that a little bit better by the end. And to get there, we need to go back to what Gwyneth was talking about. So pretend I'm Gwyneth now, pretend I'm giving a better talk. And right, we've got a whole bunch of neurons that are detecting a looming stimulus. Let's pretend there's 50 of them. I don't know, is that roughly right? 50? Okay, it's not off by too much at least. So we've got 50 neurons that are talking, that are telling these other sort of two magical neurons that are listening, right? That are going to make you jump forwards or backwards. So we've got 2 listening neurons and 50 talking neurons. And what Gwyneth was able to do, which is pretty remarkable, is figure out how strong each of those connections is, right? And she was able to then describe that in terms of a matrix. And it's color-coded, right? So like red would be like strong and positive. For her, blue was weak. You might even, in some networks, blue might even be strong and negative. So let's just pretend we're going from strong and positive with red to strong and negative with blue. And so every one of these little checkerboards is a number that like she got by counting the number of synapses, the strength of connections between neurons. So it's a very real thing, but now in this rather abstract format. And to sort of obey certain mathematical conventions, I'm going to flip it on its side. We've got 2 listening neurons, so that's 2 rows. We've got 50 talking neurons, so that's 50 columns, but I only drew 8 because like 50 was too much. Okay, so where am I going with this? Well, you could also describe the activity of the neurons also as a list of numbers, 50 numbers. How active is each neuron? Is it really active because the thing's looming over there, or is it not so active because it's over here? So let's say that black is really active and white is not very active, okay? So if we want to know what those 2 listening neurons are hearing, it turns out it's just the same linear algebra that they would teach you in 12th grade or first year of college, depending on when you took this stuff. And we won't go through exactly, but you know, you can ask me during the Q&A if you want me to define, but there's like a well-defined rule for how you multiply 2 matrices. And you get something really simple out of this. You multiply these 2 matrices, you just get a new tiny little matrix with just 2 numbers in it, right? Well, what do those 2 numbers tell you? They tell you how much each of these neurons cares about how much each of these neurons' activity is going to change in response to what it's listening to. So maybe black would be that neuron's activity is now going to increase because it's getting a lot of upstream drive, whereas like white is maybe like it's not getting a lot of drive, so maybe its activity is staying the same or maybe even going down. So that's all well and good in the system Gwyneth was talking about. A lot of the brain areas we care about, though, have this kind of wacky feature that instead of just a bunch of neurons that talk to neurons downstream, all these neurons also talk to one another. And we think that's actually a large part of how they do their computation. I talk to you, you talk back to me, I might even talk to myself. I talk to Janet, she talks to Jane, Jane talks back to me. Activity kind of flows around in a circle, which sounds complicated, and it is, but in the end, it's not. So you can describe these connections in the same way that Gwyneth did. We've got 50 talking neurons and 50 listening neurons. They're the same neurons, right? The same 50 are listening and talking. But we can perform the exact same trick. We just have a matrix of numbers that describes how strong each one of those connections are. And then we have a vector that describes A vector is just a name for a list of numbers, right? We have a vector that describes how active each of these neurons are. And we're just going to do our linear algebra. We're going to multiply our vector by our matrix, and we get a new vector. And this just tells us how each neuron's activity is going to change in the very near future. So again, if it's on the darker side, maybe it's going to go up. If it's on the lighter side, maybe it's going to go down. And something I think very subtle but super important here, right, which is Gwyneth can like literally get this, right? She can go in and measure actual physical connections and then turn them into this abstract format. I can't do that, right? Because I'm not going to kill and freeze and slice up my monkey's brains, right? And even if I were to do such a horrible thing, there'd be like 50 million neurons. It would be hopeless. So I have to somehow guess what this matrix is from data alone. And that sounds impossible, but it's really not. Because you can sit there while they play these games for hours, and what that means is you get to observe many, many, many different patterns of activity, and you get to observe all the ways in which activity changes right after every one of those patterns, right? Every 10th of a second, you see a new pattern and a change. With all of that data, you can make a very good guess as to what that matrix looks like. It will not be perfect. It will not be as good as Gwyneth's, but it will be, for present purposes, good enough. So I've got to learn this. I've got to approximate this. Okay, so now to wrap up, what does this help? It's the last slide, but it'll be a long slide. So, hold on. So how does this help us do what I was talking about earlier, right? How does this help us figure out what the neural activity is doing? Well, there's really two components. I need to be able to guess what neural activity is right now. And if I can do that, it's actually pretty easy to do the second part. I won't get into it. If I'm sure I know what neural activity is doing right now, I can make a really good guess as to what the monkey's trying to do. And we're just going to put aside the second problem. The hard problem is figuring out, especially from noisy recordings of a tiny percentage of all the neurons in the monkey's motor cortex, we're trying to guess what neural activity is doing right now. So up on the top there, right, there's that sort of weird cartoon that you saw before. Yeah. And what that is neural activity. Imagine if you only had 3 neurons, right? You could plot the activity of all 3 neurons in a 3-dimensional space. You don't. You have like millions of neurons. So you would need a 1,000,000-dimensional space, or maybe 200 if that's how many neurons you measured. But it turns out there's tricks where you can kind of pretend that there's 3 important neurons, sort of magical, sort of proto-neurons, and those are the 3 neurons that matter the most. We won't get into exactly how that works, but that allows us to make a 3-dimensional plot of what neural activity looks like at any given moment. And that's also going to make our connectivity matrix really simple. Right now it's just a 3x3 matrix again, easy peasy. So suppose you knew for sure what the brain was doing or the motor cortex was doing right now. Well, remember, if we multiply it by that matrix, that tells us what the change in activity will be. Well, if you know what it's doing and you know how it's changing, you've got a pretty good guess at what it's going to be at the next moment in time, 20 milliseconds from now, let's say. So you got a pretty good guess, but it won't be perfect. But that's okay, because you also had data. You also are making measurements. They're also deeply imperfect, so you don't entirely trust them. But what you can do is combine those two, and you basically end up taking a weighted average, and that is your absolute, mathematically provable best guess as to what the brain is doing right now, okay? And then, assuming that everything is going well, you can just take that, that's now your best guess, and you just run the process forward. So as long as you believe that you're doing pretty well, and you're getting reasonably good data, you can continue making very good guesses sort of essentially forever. And this isn't just something that you would use for this kind of an application. When your GPS is updating as you drive down the freeway, it's doing the same thing. When they're guiding airplanes, very similar math is being used. You often have noisy sensors along with some notion of the physics of how things change. So in any case, this is what's allowing us to get a very accurate estimate of what that little green dot is, which is the state of neural activity in this kind of fake squished down, pretending there's only 3 neuron space. But it's good enough for us to be able to make a good guess at every moment. Is he on that purple path and going forwards? Is he on orange path and going backwards, or is he down at the bottom and stationary? And you can see by looking at the right that the actual and decoded velocities are, amazingly similar. And so this works very well. So I'm done. I just want to thank the people who did the work, and because you can never get them all in the same room at the same time, we're going to have to shuffle through a few different photos, and thank you guys very much. And I know we're going to be up there taking questions soon.
All right. Thank you so much, Dr. Churchland. I will now invite both Dr. Card and Dr. Churchland to these chairs here, our stage, so that we can have an interactive discussions about the questions you may have. All right. So as I said, we've had some questions come in online. What I'm going to do is alternate between questions from our audience here and from the online questions that we've received. Just to start off, I guess I'll ask you a question that I think has come up multiple times in our online questions as well, and I think it might be good to get us all warmed up about it a little bit. We heard two great perspectives from physiology and also from mathematical perspective about addressing a common problem. What would you both think, from what you've learned so far, would you say that a single neuron might be responsible for a very specific behavioral output, or would you rather say a single circuit is responsible for a single behavior? From what we know so far, what would your insight be?
So I think that's a great question for us because we do come from these two different perspectives. So I'll represent fly perspective. In the fly, I think it's both. So we clearly have many individual neurons that are identifiable by how they look and that do very specific things. So I showed you a couple of the descending neurons, which when we activate them, make the fly do a very precise behavior. So there clearly is a role in many cases for individual neurons. At the same time, there are other parts of the fly's brain that clearly act more like a network. So there are areas, these names might not mean anything to you, places called the central complex or the mushroom body, where there are more recurrent circuits that are a little bit more like what Mark talked about. So I do think we have that range in the fly.
Okay, that's great. And it would also be context dependent, obviously, depending on the kind of stimuli you receive.
Absolutely.
Yeah, of course.
Yeah, and at the risk of agreeing too much with Gwyneth, yeah, it's the same in the monkey that you can see, and maybe more importantly, right, in the human, which is ultimately, where we're all trying to get. A simple example of something that's very dedicated, you all know, right, is the stretch reflex, right? And there's a very dedicated set of circuit, and it's such a simple circuit, it's really only two neurons. One neuron that does the sensing of the stretch and goes into the spinal cord, and another neuron that comes back out and makes your leg do this. And it's not for silliness, right? It actually helps stabilize you when you're standing. So there are these very dedicated circuits that create very specific behaviors, and that tends to be particularly true for things that are evolutionarily sort of very old, and you want them to be very hardwired. And kind of like Gwyneth was saying, there's also these other parts of the brain, and in us, motor cortex, it's the principal thing that moves the arm, and that circuit is not sort of pulled apart into different subcircuits, right? Every neuron in that circuit will be active for basically every different skill, right? It won't be like there's some neurons for baseball, and other neurons for hockey, and other neurons for coffee cups. It seems like it's a very all-purpose circuit that can somehow, as a whole, learn to do a lot of different things. Yeah.
If you would like to ask a question, please raise your hand, and Olivia will come to you with a microphone. Right over here in the front. Oh, sorry.
Hi, I'm one of the teachers in the audience, and my question is probably for both people because you study mammals and you study the looming behavior. So I used to live in Florida, and when you drive in Florida, there is always a squirrel, series of squirrel on the side of the road, and for some reason they do not follow their flies. They do not go away. They just go under the wheels. What do you think causes the behavior? Because you know.
No, it's a great question. So that actually gets at a deeper scientific issue here. So what I talked about today was this sort of pure looming reaction in flies. But actually where we're going in the lab now is we're trying to complexify the fly's environment, because of course, flies rarely in a totally white room seeing a dark stimulus, right? It's gonna have a leaf it could hide under, and so what does the fly do? And these are studies that we're actually modeling after ones in mice, so this is true of mammals too. There seems to be at least two very different strategies that animals have. Maybe you could count three. So one is move away from the threat. One would be freezing. Then the other one would be move towards some kind of goal. It could be shelter. It could be that the animal thinks it can be faster than your car moving, but it has some goal and it's kind of a positive thing it moves towards. And so this has become a really interesting thing to study from the sort of movement choice point of view, because the brain is clearly doing these different computations at the same time, right? The goal versus the aversive computation. And how do those get combined and how does it get chosen between? Clearly the squirrels, are choosing one more than the other. Okay. If you guys want to think about some questions that you might want to ask, is there somebody? Okay, I have.
Hi, thank you so much, both of you, for this amazing talk. I really wanted to direct this question to Dr. Card. Specifically, I noticed that there were a lot of different outcomes for the fly. Could have, yes, you had a correlation in which it moved a little forward or maybe that, but some of them completely went the opposite direction in some way, shape, or form. And so that wasn't necessarily uniform. So even though there was looming stimulus, I wonder, what do you think might have triggered a different response in terms of maybe misperception, or do you think it was environmental conditioning or behavioral, maybe a combination of both?
Great question. Again, kind of spot on for where we're trying to take this work. So there are a couple different reasons we think we could see this variability, right? So the stimulus comes from the front and yeah, maybe on average it jumps backwards, but there was also the fly that jumped left. Why did it do that? One is simply noise of sort of the initial conditions. So you can imagine our flies might have been surprised while they were trying to groom. They might have already been kind of hanging out a little bit on the side. And even though they try to do these postural adjustments to get to these target locations, they might not have the time to get all the way there. So they're kind of doing the best they can with where they started from. So that adds a little bit of noise and variability. But there's actually more to it than that. So we have now done repeated studies. So in our current setup that I talked about today, it's sort of one fly per looming stimulus. We show the fly looming stimulus, it flies away into the world, and we have our video, and we do the next one. But now we've taken the same fly and shown the same stimulus again and again. And what we see is that individual flies actually sort of have habits, right? So a fly that chose to go in a particular direction or make a particular behavioral choice on trial one tends to repeat that behavior. However, that habit, or sort of bias, if you want to call it, only lasts for maybe a couple hours. And then if you take that fly and you test that same fly again on day 2, it now has a different choice. And so what we think is that there are these transient states that maybe last on the scale of hours that are really internal states, right? Maybe that fly was hungry, or maybe that fly had some kind of different exposure in the vial that it was raised on, and some different encounter before it came up. And so these internal states really do seem to have a big effect on the choice, and we're trying to get into that now.
Thank you for that question. So we have a couple of questions coming on online about new technologies that we hear about in the news a lot, and some very controversial ones, and people want to know your ideas, thoughts on those. And so one is Neuralink. So what do you think, is there any, is there something particularly exciting about it, or what do you think about Neuralink specifically, or any other technologies that you might want to tell audience about?
Yeah.
Yeah.
Yeah, this is one where, being careful is, I think the basic technology that's being developed by Neuralink is potentially really powerful and could really benefit people. The current state of the art, if you're a human, and very small number of humans have had what they call BMIs, or brain machine interfaces, or sometimes BCI for brain computer interfaces. implanted in them, and solely for experimental purposes. But it can work very well, and there are videos of truly impressive things, like people being able to render handwriting or render speech or type at pretty remarkable speeds. People who are very impaired, who can barely move, but they can do it directly with their brain. So it's something that really has the potential to help humans. It's currently limited by, current technology would be a lot better if you could record from many more neurons stably over a lot longer period of time. And the neurotechnology absolutely has the possibility of giving us that. At the same time, I think we all have to be incredibly careful. I mean, if you're going to be putting these things in people, you have to be very thoughtful, very careful, very sober, and about what it is you're going to do. And you know, these things need to be thought through very carefully, and one should be very careful not to overstate what things can do. So yeah, those would be my thoughts. Yeah.
Just because you can do it doesn't mean you should do it.
Yeah, yeah, I mean, there's a lot of issues. In the end, most of these issues are obvious. If you got a bunch of reasonable people together in a room, they would come up with the right answer. It's not that hard, but it is important to do, and it's important not to yeah.
Anybody else in the audience like to ask a question before I move to somebody from them? Okay. Yeah.
Amazing talk and wonderful graphics, just very memorable. So I teach teenagers who are in a transfer school who, many of them, we find if they stay with us long enough, have substance use issues, right? We're not gonna call it abuse, who are we? Use. And I'm listening to your talks, and I'm wondering what your thoughts are about how to create muscle, maybe some kind of cognitive, muscular, see I'm forming this as I go, kind of therapy for young people who have the habits that are associated with use, right? The physiological, right? I don't want to use anymore, but I don't know what to do with my hands and my mouth, right? Those are very real, complex concerns, right? They don't want to use, but they have the embedded habits. What are your thoughts around the kind of work you do and how that could be in some better future associated with some kind of treatment modality for that purpose?
Yeah, yeah. I think it's a great question, and I won't have an equally great answer. But you know, in both the fly and in primates and in rodents, there are these very well, I wouldn't say they're well understood, very heavily studied circuits where if you do something that gets you a reward, and a reward could be food or water if you're thirsty, could even be something like friendship. You know, most of us know dopamine is released, and there's in ways that we don't fully understand that causes you to then want to repeat that action, to repeat that outcome again. And we do know that drugs of abuse hijack that pathway in bad ways. The motor system is heavily intertwined with that pathway, kind of for the reasons you might expect. And so it's not just about decision making, it's also about movement. But we don't really understand very much about that yet. There is interesting ongoing research, but I'd be overselling it if I claimed that we know a lot. There are a lot of people who are working now on better therapies for drug abuse, and then there's those of us who are trying to understand the basic neural circuitry. And you know, one hope is certainly that if you understand that better, a variety of therapies might present themselves, unexpected possibilities even, but you know, I think we're looking 10, 20 years down the line at that point. Yeah.
Just to link that a little bit on the sort of the basic research front, I think that is one of the things we're hoping down the road a long way that the fly could contribute to, right? Because the fly is a place where we could unpack these conflicting motivations and these conflicting desires that are clearly feeding into these decision processes that we want to try to influence, but we don't really even know how they're coming together, right? So if we can come up with models and whatever organism where we can start to understand in a really detailed mechanistic way how those things are getting integrated, then yeah, the hope is that one could start to understand therapy.
Hi, so I'm a teacher and I teach the science of emotions, and we're recently talking about like the effect emotion has on behavior. And so the whole time, Dr. Card, I'm listening to you talk, and you're talking about these flies either flying forward or flying back. Now, my fly anatomy is a bit rough, but the one question I'm asking myself, so for humans, the response is fight or flight or freeze, right? So with this looming stimulus, maybe this is a basic question, how come, I guess, the behavior isn't similar? Or why is it in each instance, the fly is either going forward or back, right? Where is the freeze? Or where is like the other option, I guess?
Yeah, no, that's a great question. So the answer is that it actually is very similar. I didn't unpack for you all of the things that we see the flies do when we show these looming stimuli. I think we've literally looked at 30,000 flies now. And today I talked about the subset of those that do the flight. So yeah, so we were kind of focused on the flight part, but absolutely, freeze is a significant part of it. Now fight, we've never seen a fly actually stay to fight the looming stimulus. But flies do have aggressive reactions to each other, especially the males have these aggressive displays where they might either leave the arena or stay and engage the other male. And so that would be a place where I think you actually find very parallel behaviors.
If you see videos of flies fighting, it's actually kind of remarkable. Yeah, no, I'm serious. It's worth looking up.
Yeah, definitely.
Yeah.
YouTube it.
Yeah.
Okay, so we have an interesting question that came in online. What are your thoughts on creating a fully functional digital map of the brain? And maybe this is something you can talk to about with the EM data set as well. Do you think that's like a representation of the dynamic system in the fly brain, or do you think it's like a snapshot of a singular moment? What do you think?
Yeah, it's a great question because there's an ongoing discussion of, should we scale this kind of connectome project up to larger brains, right? It's actually a significant investment of resources, especially as the brain gets bigger and bigger. And as you point out, there's no dynamics in it, right? There's no change over time. There's no learning. You can't study changes in these synaptic weights in a connectome. But I think at least from the fly example, and not just my work, but across the fly field, I think what we've really found is that it's a starting point for studying all sorts of questions about the brain that now that we have it, we realize we couldn't have done without. There's just things that are in the architecture that you can see from these connectivity patterns that give you clues as to how to study things functionally, that without that physical substrate, it would be very, very difficult to deduce from experiments alone.
Do we have somebody from the audience who'd like to ask a question? Please do introduce yourself as well.
Hello, my name. I'm a student in 11th grade. I was just wondering, if you had a situation where you had basically like a absolutely identical test subject in the same conditions, same everything, would there be an absolute only one reaction, or would there be multiple that could possibly happen, like there's multiple I don't know how to say it, multiple reactions to that stimulus? And if there is one absolute reaction, is it possible to create maybe a virtual brain or virtual network that can basically respond how any given creature would?
Yeah, great. Do you want to dive in? Or
Sure, yeah. So yeah, I mean, this relates closely to the earlier question about variability. And an interesting hallmark of human and really all animal behavior, kind of as Gwyneth was already discussing, is variability, right? The same stimulus doesn't necessarily always lead to the same response. And we all take that for granted, right? I don't, is there a sport you particularly like to play, especially any that involves hitting balls?
Unfortunately, no.
Okay. Well, we're all very familiar with, or maybe an instrument, right? We're all very familiar with attempting the same act multiple times, and you might succeed 5 times in a row, and we know that doesn't mean you'll succeed on the 6th time, right? And if you think about it, that's really weird, right? I mean, if you built a robot that, say, did free throws, and you dialed in that robot such that it was hitting swishers every time, right? It would never miss again. I mean, not unless, like, you know, the temperature of the air changed a lot or somebody moved the hoop, right? So it's a little weird that you would think once you had succeeded a handful of times, you would always succeed, and yet it's not like that. And we kind of actually know the answer, or at least strongly suspect we know the answer, which is that, you know, our brains didn't evolve to deal with situations like that, right? They evolved in a world that's itself fluid and variable, and you never see the same thing twice. So what they're really good at is coming up with a pretty good movement or a pretty good answer in relatively novel situations, right? You're a cave person, you're trying to hunt your dinner, you've never stood in that exact place with that exact spear, throwing it at that exact prey before, and yet you have to do something that stands, you know, hopefully better than 50% chance of getting you dinner. So it's as if the brain resolves the problem every time it sees it, rather than going back and just getting whatever worked last time. And we can actually see that happen inside motor cortex before movements start. We can see the variability of the brain coming up with slightly different solutions, and it then predicts small variations in the movement itself, you know, half a second later. So we actually can trace that variability inside the brain.
Okay, so we're sort of, okay, wow, we have more questions. Yes, right there.
Just another thank you to both of you. This is a great pairing. My name's Ivan Amato. I'm part of the communications team here at the Zuckerman Institute. Just kind of interested in the synergy we see here between, say, this anatomical approach to understanding a circuit that goes from sensory information to movement, and then this more computational mathematical one. You know, and here you are two working together. So I'm wondering how, Gwyneth, when you're getting this mindset from Mark, how that's changing the way you're thinking about what you do, the kinds of questions you ask, and the other way around. Is working with someone who is looking at this detailed anatomy, does that change the way you think mathematically for these kinds of problems?
Yeah, this has been a great experience for us because we've had some really fun conversations very similar to what you're asking. I think what we're really interested in doing and kind of taking some of Mark's perspective is now understanding the context of activity in our circuits. I mean, I talked to you about basically 3 cell types, right? A looming detector and then like a forward and a backward neuron. But even the fly brain has 300,000 neurons. So what is the population activity surrounding the activity in that particular circuit where we know the function so well, but now can we see context? Can we see representations of other things in the world or representations of the fly's internal state and how that's influencing this circuit? So it's really useful to try to bring this sort of broader and sort of more abstract computational perspective to these specific circuits we look at.
Yeah, I mean, I think ultimately, you know, we hope, or maybe even we insist that it all link, right? That, you know, in the end, it's just physical stuff. I mean, your brain's just made of meat, and yet it thinks and it moves. I mean, and you know, all of this. I mean, you saw the incredible detail in those cryo-EM images, right? And you know, all of those intricate connections shape how activity flows, and how activity flows performs computations. It makes decisions, it generates movements, it absorbs perceptions, it instantiates emotions, right? It does all of those things. And so there's no way to understand this all in some sense without understanding how connections between neurons produce certain patterns of activity and why those patterns of activity are the right patterns of activity for doing that particular computation. And we might focus more on sort of one end of that spectrum or the other, but it's got to all hang together or else something's wrong. So yeah.
I think we have time for one more question. You had your hand up before, yes.
Good evening. Thank you again for this lecture. I'm a special education teacher. And one of the things that I teach my students is this connection between mind and body. So for example, if you're learning about something new and you take notes on it, you tend to remember it a little bit better. And I'm curious, it sounds almost like, it sounded so almost overly simplistic that the motor neurons fire through the motor cortex, go straight down and activate that muscle. So what is the connection then to memory? Is there like a separate pathway that's connecting to the rest of the brain? Is it something we don't fully understand? I apologize too if this is out of your area of focus, but I'm curious how muscle movement and motor neuron activity can translate into memory.
Yeah. I mean, probably the sorts of phenomena you're interested in some sense, would happen in the brain areas and the neurons that are actually just upstream of motor cortex. So, you could think of a motor cortex as a machine for generating movement. It's a very flexible machine that can generate a huge variety of movements, including new ones that you learn in your lifetime. But it's got to also take its instructions from the parts of you that make decisions and have beliefs and choose to act in accordance with your beliefs and things like that. So I don't think it's the motor cortex itself, its activity, that would be helping to ingrain the memory. It would more be it gets a little bit tricky because sort of like thinking and moving, they kind of mush together, right? Like cognition and motor control. We like to pretend they're separate, but they definitely sort of form a continuum with one another. So that's a slightly rambling and not great answer, but that's, yeah.
I'll add a little bit too, from like the sort of maybe the simpler framework of the fly, right? Which is that at some level, memory is just that synaptic weight matrix, right? So whether it's the fly, color coded that you saw or Mark, if that changes, you're going to do a different action. And that can change, like imagine if the fly sees 5 looming stimuli in a row, we actually know that habituates its response. You can imagine those colors decreasing. That's effectively a memory, right? Now, whether it has consciousness that like can recall it as a memory, but you've changed its behavior in response to a previous experience. So at some level, that's I think what we liked so much about that weight matrix.
Yeah.
It encompasses so many of these concepts.
Yeah. No, thank you for rescuing me, Gwyneth. I think what she said is just really critical. So critical that maybe we should have said it out loud 3 or 4 times, which is that in some sense, the fundamental theorem of all of neuroscience is that it's the strength of the connections between neurons that get the job done. And so any memory is a change, and it could be a subconscious memory, it could be a conscious memory, it could be a motor memory, it could be a memory of your childhood, but we fundamentally believe all of those are changes in that weight matrix, right? Just, they might be dramatic, they might be subtle, but those connection strengths are changing. And there's a large field, including people in this room, that studies that, how those changes are made and how they're kept. Yeah.
Great. I think that's all the time we have for questions today, though this has been so great and engaging. We should have more time for questions. But as we wrap up, I think I'd just like to add a closing question, or maybe closing remarks would be a better term. So as we all walk out of this room, I'm sure we're going to have achieved much greater appreciation for how much goes into every single little movement that we make. As you saw from Gwyneth and Mark's images of neural circuits and how well orchestrated it is to actually give out a specific motor output. But my question for you would be, was that with all that you know so far, what is the next research question that you're really excited about answering, or would really like to unravel next with the wealth of information that you have and the tools that you have? What's the next really thing that really excites you that you could help unravel?
So I think for me, the audience basically already uncovered it, which is that, I talked a little bit about a fixed system, and the question is, how does context, how do different states, how do different, ways that things that animals experienced affect these circuits, and how does it change them?
Yeah, I mean, my answer is somewhat similar, and we were talking about it earlier, sort of during I'm not sure if it was prep, or I think we were just having fun and chatting, but we were remarking on the fact that in the real world, you can make a movement you've never made before in a context you've never made before, and primates, and especially humans, are unusually good at that, right? And it's one thing to understand how the brain generates a highly trained movement that an animal has made, many, many, many times and is really good at. But how can we encounter a completely new situation and do something pretty good in that new situation? I think so far our field really hasn't tackled that problem yet, but I think we're close to being able to. Yeah.
Exciting. Thank you so much, everyone. That's all the time we have today. Thank you all for attending this event. And thanks again to both our speakers for joining us today. Please do take 2 minutes to fill out the survey, which we will put in the chat for the online community, and let us know what you thought about today's event. And we really appreciate your feedback in person and online. Thank you again. Thank you, everyone.
Thanks to our moderator.
Thanks, Samara.
So I'm just going to close this out. Thank you again to Drs. Card, Churchland, and Zamarad for this great event. And if you all liked this, we're going to be back in February on Valentine's Day. None of your plans. On February 14th for the next Stavros Niarchos Foundation Brain Insight Lecture with Drs. Dima Amzo, Danny Dimitriou, and Katie Insel as they discuss mental health in childhood and adolescent development. And until then, I hope you stay healthy.
That does sound like quite a date night. Yeah.
Can I get a quick photo?
Sure, yeah.